Good afternoon. My name is Alfredo Fontanini, and I'm the chair of the Department of Neurobiology and Behavior. Welcome to the 24th Schwartz Mind-Brain Lecture. And I'm particularly excited this year to be able to bring back this very, very prestigious lecture here at Stony Brook after a 2 years break, and I'm sure you can all imagine why. So I'm particularly grateful to those of you who decided to and were able to make it in person, and also want to welcome those who are following the live cast online. So the Mind-Brain Lecture has been organized by the Department of Neurobiology and Behavior here at Stony Brook University, and has been supported through generous funding from the Schwartz Foundation. Let me tell you a few words about the Schwartz Foundation. The Schwartz Foundation was created about 25, 27 years ago to support a then newly born field called computational neuroscience. This is a field that sees the confluence of neuroscience, biology, engineering, computer science, physics, and mathematics. And the goal of this field is to form a theoretical and mathematical understanding of how the brain works. And the Schwartz Foundation has been truly instrumental in generating this field. And in fact, till now, all the leaders in the field, all the who's who in the field of computational neuroscience was either supported, is supported, or has relationships with the Schwartz Foundation. The mind and brain behind the Schwartz Foundation is Dr. Jerome Schwartz. Dr. Jerry Schwartz was the founder of Symbol Technologies, the company who basically commercialized the laser scanning system for the barcode. And he's a physicist and an engineer, and he's also a friend of the department and of Stony Brook. In fact, he sits on the board of trustees of the Research Foundation. So this lecture, and Dr. Schwartz wanted to thought of this lecture as a way to bring very high-profile speakers on campus and expose the community, both of students and beyond students and scientists, to themes related to neuroscience and related to computational neuroscience. So this year's speaker is Dr. Donald Katz. Dr. Katz is a professor in the Department of Psychology at Brandeis University and is a member of the Volen Center for Complex Systems. He received his undergraduate education at Brown University, his graduate education at Indiana University, and he had his postdoctoral training in the Department of Neurobiology in the Medical Center at Duke, sorry. Don Katz has been a true pioneer in the study of taste and in study of the neurobiology of taste. Prior to his work, the field was mostly focused on understanding how molecules would bind to receptors on the tongue, and was actually paying very little attention to how the brain builds the gustatory world. And in fact, Don Katz's work single-handedly revolutionized the field by putting at the center of the stage the neural dynamics that are composed by interactions of neurons in the brain that really give rise to the perception of taste. In fact, the current paradigm of how the brain elaborates taste information is likely credited to Don Katz. So he has numerous publications on very prestigious journals. He likes to shy away about naming those journals, but he published on Cell, Neuron, Nature Neuroscience, PNAS, Journal Neuroscience, and so on and so forth. He's received multiple awards, the Ginomoto Awards, the Pollack Awards, and he's received also several teaching, mentorship, and educational awards. And this reflects the fact that he's an incredibly talented educator and is very, very committed to education. So today, Dr. Katz will tell us about his work on taste, which very nicely blends a computational perspective with a behavioral perspective on understanding how the brain builds taste. And the title of the talk is Constructing the World of Taste in Your Head. Please join me in welcoming Dr. Don Katz, the 24th Schwartz Mind-Brain speaker. Thank you.
Thank you, Alfredo. That was really very nice. Thank you, Alfredo and Kathy, for taking such good care of me today. I'm grateful to the Department of Neurobiology and Behavior and the Schwartz Foundation for bestowing this honor upon me and giving me the chance to talk. I'm grateful for those of you who've come out. I know what the last 2 years have been like, and it's nice to see you here. It's also nice to know that there are people online. Hello to you as well. So I'm going to talk to you today about constructing the world of tastes in your head. But so to start, let me tell you that a kestrel can see a 2-millimeter bug from the distance of the top of an 18-meter tall tree. And what that means is that this bird can see the average American baseball from the distance away of not just one average American football field, but 6 football fields. And so the take-home message here is that vision can be you can see things very, very far away. And so if you think of your skin as being here, a visual stimulus can be detected very far away. Far enough away that the truth is you don't have to do anything about it. An auditory stimulus, something you hear, you can also hear from very far away. Perhaps not as far as what you can see, but given the complexity of our visual environment, a lot of times you actually can hear it even before you see it. It's also possible to smell things at a certain distance. It's usually possible to tell who dealt it from at least across the room. Furthermore, the sense of touch, while it is actually against your skin, is still an object that is external to you. But it's a lot closer. And so you can see that the sensory systems of vision, audition, smell, and touch have this range of distances involved with them which are associated with different immediacies. If you have something touching you, it is fairly immediate that you have to do something about that. You have to actually figure out how to act in relation to the thing that's touching you, generally pretty quickly, whereas something that you see, in general, you do not have to worry about in the immediate sense, and a lot of times you will have no reaction to. But there is one sensory system left out of this, which the situation is very different and quite a bit more dire, because the sense of taste occurs when something is inside your body. You do not taste something until it is past your skin, in your mouth, and that means the immediacy of the sense of taste surpasses that of any other sensory system. Put simply, when you have something in your mouth, you must make a decision what to do with it. You must. And so what I've got here are videos of little rats that are used in our experiments. And what you're about to see is what this rat, the decision that this rat makes as to what to do when a small aliquot of sugar water is put in her mouth. And if you look closely, what you can see is that she's running her tongue from side to side and pulling the fluid back in. This is the rat yum face. This is the rat letting you know that it likes what's in its mouth, and the decision that it is making is to swallow it. Contrast that to the same rat when a small aliquot of quinine has been put in its mouth. Quinine is very bitter, and it is very unpleasant, although it is delicious with gin, and the reaction of this rat is going to be very different to this stimulus. In particular, you are going to see what are called gapes, large mouth openings in which you will see the teeth, sort of a yawning face made over and over again. And what you can't see is that inside the mouth, the tongue is rolling forward to eject the fluid. This is the rat yuck face. And I'm going to focus mostly on this face because it's really easy for neuroscientists to measure. But I want you to know that everything I tell you about the yuck face is also true about the yum face. And so, if you want to know how this works in the brain, how do you study that? Oh, before I say that, I should mention to you, just as because sometimes people are like, well, who cares about rats and mice, that human beings are no different. A human being that puts something sour or bitter into its mouth will make this classic gape face, at least an infant will, and these faces come back under most forms of dementia. The only reason that you guys don't make the rat yuck face when something nasty is in your mouth is because you've been socially trained that it is inappropriate to do so. But in general, this is a very cross-species phenomenon. I could show you examples of toads doing this. Pretty much any animal that has a tongue and a mouth produces these faces. So how would a malcontent, by which I mean anybody who joins the Katz lab, study the brain and try to figure out how this works? So a little tiny primer on basic neuroscience. The brain is full of neurons. And this funny-shaped object is a standard neuron, of which you will find in the brain. And what we tend to do is we will insert electrodes into the brain so that we can record the electrical activity of these neurons. And this electrical activity takes the shape of sequences of spikes, action potentials they're called, voltage spikes that happen over and over again. And since they all look alike from the same neuron, you can shortcut them. And that is a representation of what this neuron is doing in response to some stimulus. And so you can see that it is a sequence, almost a Morse code-like sequence of pulses. And now, what's standard in the field is to take these pulses, add them together, and use that single number as a representation of what that neuron is doing. And so one of the basic questions that we asked, one of the basic approaches that we take in my lab, which is dedicated to questioning the status quo in science, is this the right way to go? Or rather, does time matter in the processing of tastes? Does it matter not just that there are 3 action potentials here, but that they happen in a rhythm, boop, bop, boop? It's also important to remember that this neuron is not alone. It's sitting there in the brain surrounded by other neurons. There are billions of them up there, and they are connected to one another. And so when one spikes sense action potentials, those action potentials impact the other neurons in its vicinity, impact some of the other neurons in its vicinity, potentially causing them to fire action potentials. And so the second question I want to address in this talk is, do neurons speak with one voice as they process tastes? It's standard practice to just talk about each neuron separately. But we question that and ask whether you learn something more when you consider the neurons as a group and if they are in fact working together. Not just bunches of neurons all firing, but neurons doing something as a functional unit. And as long as we're talking about functional units, it's important to remember that these neurons are in brain regions. And so this is a little tiny drawing of the taste system. Each one of these dots is another brain area full of neurons, and they're all connected to one another. So the same question that you ask about neurons, you can ask about brain regions and say, do brain regions interact during taste processing? And if so, how? One word got cut off at the bottom. And so while I'm going to talk to you about taste, we are approaching these 3 big questions in what we do, using behavior, neurobiology, and computation. All right, so dispensing with the first question right immediately, I'm going to do something that always strikes me as bittersweet. I'm going to reduce my 5-year postdoc at Duke University to a single slide. What we learned at Duke is that, in fact, time does make a difference, that it is not a good idea to describe the responses of neurons to tastes with one number that's an average of spiking across time. And so let me explain this graph, which is a standard kind of graph that is used in the study of taste. We have what is called PSTHs, or peri-stimulus time histograms. Along the bottom, we have time where 0 is the time that a taste was delivered to the tongue. And so we can see what happens afterwards. In the y-axis is the number of action potentials that were produced across a particular unit of time. And so what you can see right away is, what you've got are overlain PSTHs, overlain responses of this particular neuron to a whole set of tastes on the tongue. And for now, I don't really want you to worry about the whole set. Just notice that one of them is really yummy, that's sucrose. One of them is really horrible, that's quinine, and the others are sodium chloride that are varying degrees of somewhere in between. And what you can see from looking at this is that the responses change through time. And in particular, when we did a whole bunch of work on this, what we found is that you can think of the responses as happening in 3 successive epochs. And the first thing that happens when the taste hits the tongue is that neurons respond with a burst of action potentials, an increase in firing rate, that is non-distinct. It's the same to every taste, and that's why each one of these peaks is about the same. And so this we call the detection response, which is to say that looking at this graph, you can detect that a taste has hit the tongue because there's this sudden burst of action potentials, but you can't tell anything else. But fast forward just a couple hundred milliseconds, and now the responses to different tastes are different. And so in this period that we call the identification epoch, you can now see the difference between the firing to this taste, the firing to this taste, the firing to this taste. The neuron is giving you a taste-specific response after a couple hundred milliseconds. And then if you look beyond that, you see that the responses change again, and out here, now the response has taken on a special aspect that it didn't have before. When you get out here to around a second after the taste has hit the tongue, this neuron, you can see, is firing a whole lot to the nastiest taste, quinine, and is firing nothing at all to the best taste, sucrose. It turns out that what this neuron is doing is firing in a palatability-specific way. It is telling you what the taste is to the animal. So to put this in a psychological perspective, in this neuron you can see the animal going, hey, there's something on my tongue. Whoa, it's quinine. Ew, get it out. And in fact, this change into palatability happens at around the time that the animal makes that gaping response. And in fact, we can do very simple analysis. I'm going to keep away from going into heavy discussion of the computation through this talk. But we do use a lot of heavy computation to come up with the fact that this is palatability-related firing. We do a very simple analysis, which we correlate the firing of the neuron to each taste to the known palatability of each taste. And we can do that in any number of ways. We can talk about how much the animal drinks of a particular taste. We can talk about how many of those different types of faces it makes. But when you do that, you can see that the firing is not palatability specific here, but then it slowly becomes palatability specific as you get into that late period, as the animal figures out what it thinks of the taste and what it wants to do. And so what you can see in these neurons, which are in the primary gustatory cortex, which I'll call GC, you can see the process of figuring out what the taste is and the immediacy of the need to go beyond that and to figure out what you want to do with it. So, this basically tells you that there is at least information in the time course of the firing that goes beyond what the classic idea of just averaging across the firing and saying this is how many spikes were fired. And so the dynamics of the neuron matter. But we can see this more. The dynamics really open up when we do a different kind of analysis. And so what this is, what you had before were different PSDHs to different tastes. Here you have, with one neuron, here you have all 10 of a set of recorded neurons and a single response to a single application of quinine. And so each one of these hash marks is, as I introduced earlier, an action potential fired by that neuron. And so you can see the action potentials fired by neuron 9, you can see the action potentials fired by neuron 7, etc. And while this looks pretty hard to understand, what's going on here is there is a particular point in time when the firing changes, which is to say the transition into palatability-related firing doesn't actually happen slowly. It happens suddenly, in an aha moment. There's a moment when this neuron suddenly starts firing. At the same moment, that neuron starts firing. At the same moment, that neuron starts firing. And that neuron starts firing. And that neuron starts firing. And, just for good measure, at that same moment in time, two separate neurons decrease their firing, suddenly stop firing action potentials. And so when I put it this way, you can actually see that there is this transition point at around the right time. Again, the bottom of the slide is a little cut off. At around the time when palatability is supposed to appear, you suddenly get the rat going, aha, there is something on my tongue that I don't want there. Or alternatively, there's something on my tongue that I do want there. And so you can see this happening, and I should point out, so what we got is we've got a state preceding this, which is palatability poor. There's no palatability-related information in it. And then a state afterwards that is rich in palatability. And I should point out, because it is a computational talk, that in between these is a period of uncertainty. We have to use some fairly fancy machine learning computation in order to see these state transitions occurring. And there is a period in between when it's in one state and when it's in the palatability-rich state when we're not sure what state the system is in. And so when I have this line being a slight diagonal, it's because the analysis we're using is figuring out, okay, now the probability is high that we've transitioned into this other state. And so the temporal coding, the time-varying activity, the dynamics of this neural firing are actually highly nonlinear. There is a sudden change when most of the neurons flip into a new state together. And that new state is the animal figuring out that it doesn't like what's on the tongue, in the case of quinine, and that it wants to get rid of it. And before I show you that that's true, you might want to know, how do I reconcile the two pieces of data that I've just shown you? Because first I showed you this piece of these PSDHs where palatability correlation comes on slowly across over half a second. And then I showed you that, in fact, palatability comes on very suddenly. How do I put those together? The answer is, the secret is very simple. In each trial, the time that the system flips into the palatability-related state changes. And so what we've got up here is a set of 10 trials in which each of those trials, what I have plotted is the onset of palatability coding, the onset of the palatability-related, the transition into palatability-relatedness. And you can see that in every single one of those trials, that transition happened suddenly. But the transition happened at a different time in each trial, such that if you average across them, you get what looks like a slower ramp. And that's why we have the slow ramp down here, is because this is actually averaging across a bunch of different trials. In single trials, the rat realizes in a moment that that's what it thinks. And so, but I still haven't proven to you that what's really going on there is the recognition that something is bad. And so to do that, we actually did these recordings in the fancy computation at the same time that we took a close look at the actual animal's behavior and asked, when does the animal gape to quinine? And so here is a single trial, and this is the time in that trial that the palatability-related state suddenly appeared, and the gapes happened here, right after that onset. Here are the 3 next trials, and what you can see is that in the next trial, that transition happened earlier, and gaping happened earlier. In the next trial, that transition happened even earlier, and gaping happened even earlier. And the next trial, that transition happened later, and gaping happened later. So on each trial, that vast variability in when the transition occurred was predicting differences in when the animal would respond. And this is not unlike what happens to human beings. When we figure out what's going on, very often it's an aha moment. You're watching the TV screen, an actor comes on the screen, and you go, I know who that is, I know who it is, and it suddenly comes to you. The next actor comes on the screen, the same process occurs, but it might take a little more time, a little less time. That's what we're seeing happening here. And to summarize these data with a very old-fashioned graph, I'm going to show you a good old-fashioned scatter plot in which on one axis we have the latency of the behavior, the time that the animal starts gaping. And on the other axis, we have the time of that neural transition, that transition of the firing rates of those neurons into palatability relatedness. And the way you look at this graph, if dots, which are individual trials, fall on this diagonal center line, that means that the neural transition and the behavior happened at the exact same time. And so any dots that fall on this side of this diagonal means that the transition happened first, because the time of the transition is shorter than the time of the behavior. And if the dots that fall on this line, the behavior happened first. And so here are the data. And what you can see is that with the exception of 4 lonely trials, in an entire data set, reliably and across huge variability in individual trials of when things actually happened, the neural transition always preceded behavior by a couple hundred milliseconds, 300 milliseconds, which is really quick to us, but is a lot of time in neural terms, plenty of time for a signal to reach the muscles saying you should gape now. And so this is, we found this to be strong evidence that in fact we can see in the brain of the rat, the rat going, okay, something's on the tongue, it's quinine, oh my goodness, I want it off. And then it gapes. But this is all phenomenology. What we really have to do as scientists is manipulate the system and show that when we manipulate one part of the system, say the behavior, we change the neural activity in a way that matches, or vice versa. So I'm going to show you 3 examples of this manipulation. First, manipulate by actually manipulating the taste palatability itself. If this is a palatability-related signal, then if I change the palatability of the taste, I should change the coding. And so it's very simple to change taste palatability. Those of you who remember your freshman year as undergraduates are going to be very familiar with this. You have some tasty experience, and then not too much time later, you have a very unpleasant experience involving a great deal of gastric distress. And if you have this one experience, the very next day, the next time you are exposed to the taste of the stimulus that you previously found to be delicious, you hate it. This is called conditioned taste aversion, or CTA. And it's a very robust, evolutionarily adaptive mechanism of learning to dislike tastes that are bad for you. And importantly, for the purposes of this talk, what it does is it changes the taste palatability without changing the taste identity. When we give our rats CTAs to sugar, they can still recognize it as sugar, but it's gone from being delicious to being disgusting. And so what happens to neural activity when we do that? The answer is that it changes in precisely the way you would expect, given the conception that I've just given you. Here we have, basically these are just PSDHs, spikes per second, spikes happening on the y-axis, time related to the delivery of the taste on the x-axis. And what you can see is the dark PSDH is the one made after the animal had a CTA, and the light one is before the CTA. And here are 2 examples. And what you can see is that the first 750 milliseconds of that response are not changed by conditioning. The animal is still able to identify the taste that it was able to identify before. But the period after that, the period of palatability responding, has changed. So what this shows is that if you change the palatability of the taste, you change the palatability coding specifically and uniquely. But it does more than that, because what we've been focusing on already is that transition point into palatability coding as particularly important. And it turns out that conditioned taste aversion changes that transition time. And so in green is the average time of transition into that palatability coding before CTA. And what happens after CTA is that specific transition moves a bunch earlier as the animal becomes sensitized to the fact that, okay, I've got to figure out what to do about this taste fast. And by the way, the black line is we can extinguish the learning, we can make the rat once again think that sugar is good, and things go back. But let's move on to a second. There we have shown that, okay, you change the palatability of a taste and you change palatability coding as expected. But I want to do a more fine-grained, subtle test. What if you just change the way the animal gapes? And so a postdoc in my lab, and I'm sorry, by the way, in the bottom right corner, there is actually each time the name of the person in my lab who did the work, because of course I did none of this. I just sit around and accept big awards and give talks. The people in my lab are the people who actually do the work, but I will introduce them at the end. And so my postdoc, Jen Lee, came up with a very easy, simple, subtle way to change gaping behavior. And she did that by giving the animal a signal, a cue. And so the animal got tastes, but every time quinine was about to come, she would play a tone. And so the animal quickly learns, okay, the tone's coming, here comes quinine. And across that training, across a single session of that training, the latency to gaping drops by 150 milliseconds. All it takes is one session. Rats are not stupid. And this animal learns to gape quicker because the stimulus is coming. And just for control's sake, if you don't give the animal a cue, that doesn't happen. And so then she recorded from gustatory cortex, and the prediction was, if in fact the neural transition is what's related to gaping, if the neural transition drives gaping, then by changing this, then the process of changing the time of gaping must also involve changing the time of that transition, and that turns out to be precisely true. Across that session, that transition into palatability responding moves 150 milliseconds earlier. But one more thing, this is all from our work as psychologists. What about changing the neuron activity? What if we change that transition? What does that do to gaping? And so to do that, my graduate student Narendra Mukherjee used a very cool technique called optogenetics. What he did was, this is a microscope image of a piece of brain from a rat. This is GC, gustatory cortex here, and it is glowing because Narendra just injected a virus that causes the neurons to produce a protein that glows. But that virus also causes the neuron to produce channels that cause the neuron to stop firing when we shine a light. And so this optogenetics, there is now something in the neuron where when we turn a light on of the right wavelength, of the right color, in the brain, these neurons stop firing. And so he then actually put a fiber optic tube down to gustatory cortex so that we could do that, only we did that in subtle ways. So we had multiple types of trials. Here is the time post-delivery, and Narendra either turned the laser on for half a second, boom, boom, at the very beginning of the trial, or in the middle of the trial, or late in the trial. And so keep in mind that the time of that transition typically happens somewhere between those 2 points. And so this is going to be before, this is going to be after, and this is likely to be during the transition. And so we also did trials in which we did not deliver any laser at all, and so we can now compare in this same rat what happens in a single session under all of these conditions. And what we find is that shining the laser gives the animal trouble gaping. If you look at the graph and you say, okay, what's the average latency to gaping? And you look at the trials in which there was no laser light at all, so normal trials, the average time of gaping was a little over 900 milliseconds, a little tiny bit less than a second. And so we got the graph up here to keep track of what we're looking at, and so we're saying there is the average time of gaping. And so what happens if you deliver laser light before that time point, meaning right there? The answer is you delay the animal's ability to gape. So by inhibiting those neurons in GC for just a little while, just half a second, you screw with the animal's ability to make the behavior. More telling is what happens if you put that laser light over, when you block the actual transition, and the answer is basically the animal is incapable of gaping while the laser is on. And so this then means that if that transition doesn't occur, you don't get gaping, which means that that gaping is in fact driven by that transition. These dynamics, these ensemble dynamics between neurons that happen very suddenly, this aha moment, are actually involved in driving the animal's behavior. And of course, just for completeness sake, If you deliver the light after that, you get no effect. The animal gapes at the normal time. I should point out, though, that you might remember that I showed you a little while ago that after the transition, it takes about 300 milliseconds to get to the gape. But it turns out that even if the laser light is 25 milliseconds after that transition, which means the animal hasn't gaped yet, because it takes 300 milliseconds, the laser has no impact. Once GC has released that signal, once GC has done that transition, its job is done. It has, in fact, done, okay, something's on the tongue, it's quinine, get rid of it. And there's nothing we can do in GC to stop it anymore. And so, a quick summary of where we are now, which is about a little over 2/3 of the way through the talk, addressing the questions that I asked, 1 and 2. GC taste responses don't just code the physical stimulus. Rather, they reflect identity and palatability in successive temporal epochs. Time matters, and you can see the process that the rat is going through as it reaches the conclusion that it wants to swallow something or wants to eject it from the mouth. In analysis of single trials, that onset of palatability coding, that decision, can be seen to be revealed to happen in a sudden ensemble fashion. The single neurons suddenly change together. The animal goes, aha, I know what's on the tongue. I know whether I like it or not. And finally, this phenomenon of ensemble dynamics, of neurons working together, in turn drives the behavior. And so now, to understand how taste works, you need to understand these ensemble dynamics. All right. But we still have question 3. And this is going to be the shorter part of the talk. If neurons work together, what about brain regions? This, I should mention, is a figure drawn by a colleague of mine in Tennessee, John Bowder. And while he uses different symbols to mean different things and different abbreviations, this up here is GC, gustatory cortex. And what you can see is that these 4 regions are just 4 of the many regions that actually interact directly with gustatory cortex. Gustatory cortex sends the neural firing in gustatory cortex goes to them, and neural firing there goes back to gustatory cortex. These areas are reciprocally connected. They are constantly talking to gustatory cortex. And so it's reasonable to ask, do they help gustatory cortex do the job? Is what gustatory cortex doing happening just there alone, or are there other brain regions that are involved? And in particular, we have been focusing on a part of the brain that you may have heard of called the amygdala. We focused on the amygdala, and in particular, the basolateral amygdala, because it is reciprocally connected to gustatory cortex. Furthermore, it is known to respond to tastes, and it is known to be involved in emotion-laden decisions. And so when you have to say whether you like something or don't like something, it often involves the amygdala. And finally, amygdala is necessary for conditioned taste aversion that I've just described. And so for all of these reasons, we thought it was reasonable to suspect that if GC was working in concert with some other region, amygdala might be the place. And so we have done a great deal of work looking into that. And work that we did quite several years ago demonstrates that indeed amygdala is important for palatability. This is a graph of the palatability content of responses. And so what you can see is that when you get past 0.5 seconds, that information comes up the way I showed you in the initial PSDH. Importantly, the blue line is when the animal is intact, and the red line is when the amygdala has been pharmacologically inhibited. So in this case, we have used a drug to knock out the amygdala. And when you do that, it provides less palatability information to cortex, or cortex expresses less palatability-related information, specifically in that late time period. This is the intact, this is when the amygdala is knocked out, and this is after the drug has been washed out, which, by the way, takes a long time. And so this was an early study showing that palatability content of cortex that I've been talking about for the last 40 minutes requires or is aided by input from the amygdala. More recently, however, we've been able to do this in a way that is quite a bit more telling. My postdoc, Jean-Yu Lin, has done an experiment where what he did was he injected a virus into amygdala, and that virus carried the same things that we've talked about before. It carried a fluorescent protein, and it also caused these amygdala neurons to express channels that responded to light by shutting the neuron down. And then, over time, the connection from the amygdala to cortex takes up those channels, and it gets to the point where it also can be shut down. And what Jean-Yu did then is he actually put his electrodes and fiber optics into cortex, into GC. And so what this enables him to do is he can now shine the light on GC. He's not going to shut down neurons in GC because they do not have that channel in them. He's not going to shut down the cell bodies, the neurons in amygdala, because that's not where the laser is. All that he is doing is cutting the cord between the two. And so this is an experiment where, in individual trials, Jean-Yu is able to leave all of the neurons in the brain intact, but cut the connection between amygdala and cortex. And when he does that, what he sees is not just that the amount of palatability-related information in GC is hindered, which we already knew. He's able to show that the very transition to palatability is affected. And so when you are looking at the intact animal, you can see that, as I've told you before, there is a very sudden onset of palatability-related coding. That basically there is this aha moment that we've discussed where the neuron shifts into that state suddenly and quickly and as a group. On the trials, however, where he's knocking out the input from the amygdala, again, all the neurons are fine, but they're now no longer talking to one another between regions. That sharpness of the transition, that aha moment goes away. The animal is now no longer able to go, that's quinine and I want it off. And so in those trials, the animal's behavior is altered, the animal's activity is altered, the very dynamics of cortex that are important for behavior don't happen the way they're supposed to happen. And so these data suggest that perhaps the role of amygdala is to work with cortex to decide when that aha moment occurs. In fact, if you think back a slide or two ago when I showed you how there is a reduction in the amount of palatability-related information, you might remember that it was fairly modest. There was still palatability-related information in cortex, but what was gone was that sharp aha moment. And so the suggestion that we had was that it isn't that BLA, that amygdala sends palatability information to cortex, but rather that GC and amygdala work together to make the magic happen. That the transition, that aha moment that cortex goes through, is because of it working together with amygdala. And so what I'd like to do now, as the last bit that I tell you about taste and GC, is I actually want to present you with preliminary data. Because the whole point of science is constantly moving forward. It is, okay, what we've got there, what's the next question? And so as we speak, my senior graduate student, Abuzar Mahmoud, is working on a project in which he is actually asking the question, is it right to think of amygdala and GC as working together? And so what he's doing is he has got electrodes in both structures simultaneously, and he is recording the responses to tastes in each structure, and then he is using our computational techniques, the machine learning algorithms, to decode what's going on in each area, and he's asking if the two are working together, if they are coupled, and in fact, most importantly, if that transition into palatability-related firing that we've seen in cortex is happening in concert with a change in amygdala. And so those preliminary data essentially look something like this. This is a set of neurons, and those are acupotentials being fired by a set of neurons in cortex, is the top half, and in amygdala is the bottom half. And the yellow line is the time at which the palatability-related state suddenly emerged in cortex. And what you can see is that the neurons in the amygdala were changing in exact concert. And so the cortex was not doing this alone. It was actually doing this in concert with the amygdala. Here are the next two trials. And what you can see is that in this trial, the transition happened earlier in cortex and earlier in the amygdala. Not exactly precisely, but remember that there's always a period in there when we're not entirely sure what state it's in. And so there's always a little bit of slop. And then in the next trial, the cortex changes later and the amygdala changes later. And so what this tells us is that the amygdala and cortex are coupled. They're actually a unit. And while this might seem like not super big news to the average listener, in neuroscience, it is typical to think of each brain area as having its separate function. They are separated in space. What this suggests is that that supposition that neuroscientists tend to make is not correct in this case. That in fact, these two regions are working together to make this magic happen. Here, by the way, is a summary. This is a control condition where we show the time of the amygdala transition and the time of the cortical transition, and you can see that there is no relationship between them. And here is what's happening during taste processing, and you can see that the dots typically fall following the diagonal axis, which means that they work together. And so, to summarize this last part of the talk, brain areas don't work separately. The GC and amygdala are not doing their own thing when processing taste. In particular, the amygdala and cortex work together to process taste in a unified manner. To use the language of dynamics, they couple into a single processing unit. Palatability-related responding in cortex is enhanced by the amygdala, but more than that, the sudden ensemble change that drives behavior requires the amygdala. And the two regions work as a single object for this processing. And so how does the animal figure out what is on the tongue? How does the animal figure out what it wants to do? In this case of a stimulus being inside the mouth and a decision needing to be made, it goes through these three steps, which can be observed in population activity, that are basically the work of the amygdala and cortex coupled together, and the output of that system drives the behavioral response. And so, to summarize this in a bigger picture, the brain doesn't work the way we once thought. In fact, the massive interconnectivity between neurons and between regions ensures that the brain doesn't work in a simple way where you can think of one neuron doing one thing, another neuron doing another thing, one brain region doing one thing, another brain region doing another thing. It isn't so much a matter of which neurons do what. It's a matter of when the neurons of the group are doing what. And so, in fact, to put it another way, the question of what is the brain doing and what are a set of neurons doing can only be answered in the stream of time. You can only know what a neuron is doing at a particular moment in time in relation to other neurons in the same brain area, in relation to neurons in another brain area, and in relation to what has happened recently to those neurons. Our recordings give us a window into the processes occurring through time, and the appropriate principles to consider these are those of system theory as developed in physics, nonlinear dynamics, and interneuronal coupling. And so that is the story that I have to tell you, and I'm happy at this point to acknowledge the work of my lab, the Behavior, Learning, and Electrophysiology and Chemosensation Lab at Brandeis University. This is an almost complete list of the people who have passed through my lab in the last 10 or 15 years. And the people who are underlined were all integrally involved in the production of the talk that I've told you today. This is work that spans almost 20 years of studies, and there were about 10 or 11 studies presented in this talk. And so all of these people were deeply involved in it. I also need to send a shout out to a particular collaborator of mine, Paul Miller, who is a physicist now working in biology, and who aided us and was our consultant in all of the computational work. He played an integral role in everything that was done here. And I'd also like to thank my funding sources. which are on NIH, and particularly the NIDCD, the NSF, and cut off at the bottom, again, my thanks to the Schwartz Foundation for funding this research through the years. And with that, I am done, and I thank you very much for your attention. I'd be happy to take any questions.
Thank you for a very clear talk and very easy to follow and really fun. I was surprised that you decided to discard a piece of information that seemed to me, to keep coming back, which is the variability to that kind of flip into the aha moment, where, sometimes it's sooner than later, but you didn't seem to be interested in exploring why. Is that not important?
For those of you who couldn't hear, the question is, why don't I have anything to say about why the time of this transition happens in each trial? The answer is it's very important. And the simple answer to why I haven't addressed it in this talk is that we don't really know yet. But we are doing a whole lot of pure modeling work, which I didn't talk about at all in this talk. Basically because it would have made the talk go well over an hour. But in this modeling work, we do work that is similar to some work being done here at Stony Brook University, where we develop models that are capable of undergoing these transitions, and in fact, these sequences of changes. And one of the most remarkable things that we find is that in these models, the transitions are often driven by noise. And so one of the things that has been often discussed in neuroscience, in systems neuroscience circles, is the noisiness of the brain. And most of the models we build of brain function are such that that noise is a big problem. Our models really don't like the noise. Our models don't tend to work when the situation is too noisy. But one of the really nice things about the sort of dynamical model that makes these transitions is that noise, rather than being a problem, becomes a driver of the dynamics itself. And so when you have the system drop into a first state, or in the case of the model, a first attractor, it is the noise in the system that eventually pushes it out into the next attractor. And the reason for the variability in the timing is simply because the system is being driven by a random force. And so we do not have proof that that's actually happening in our data yet. We're working on it. But certainly, one of the exciting things about thinking about the brain in this way is that this randomness, this extra junk that everyone usually has to get rid of to make their models and theories work, actually works in our favor. Any other questions I can answer for? Yes? Okay.
So you talk about the amygdala as being very important in this, the onset of this gaping behavior, I guess. What drives the amygdala behaviorally that fits in with the kind of model you're suggesting?
Right. So the question is, for those of you who didn't hear it, well, okay, so if you've got the amygdala working with cortex, what makes the amygdala work? For starters, it's cortex. The two are connected reciprocally, and it turns out that the two are talking to one another. We now have just started the experiments in which we are knocking out the other connection, the connection from cortex to the amygdala, and while it's really too early to talk about it, it looks likely that that connection is driving the amygdala while the amygdala is driving cortex. Having said that, the system is a lot larger than just that, and we're just getting started. In fact, at dinner last night, I was asked, how can you even talk about taste when taste involves so many different things? One of the things I didn't talk about today, because again, don't have time, is that taste and smell are intimately connected. Taste and vision are intimately connected. What you taste depends on all sorts of factors. And my response was not very satisfying. Well, we have to start somewhere. And so we started with taste cortex and the amygdala, but in fact, amygdala takes input from multiple brain areas as well that are also involved in taste. We've just started looking at a part of the brain that's involved in basic feeding functions called the hypothalamus. My colleagues are working on a couple of other brain areas that are also interconnected to these regions. And The great news is that we've got careers for as long as we want them, because really nailing down this entire system is going to take many years and some additional advances in technology that enable more large-scale recordings. Yes, sir. Yes, and in fact, it is the same set of neurons, at least at the first pass. So we were first of all surprised to just find out that it's the same set of neurons involved in determining what the taste is and what to do about it. That itself was a big surprise to us. The fact that this transition, that it had to do with time and not a change of which neurons were active, was exciting. It does turn out that this transition involves some neurons going firing higher in response to quinine, and some neurons firing lower in response to quinine, and those neurons fire high to sucrose. Those latter neurons fire high to sweet. And it turns out that it looks like that transition works for both gaping and the gathering up and swallowing. But that leaves two big questions. One is, when are we going to prove that exact fact? It's more difficult to do that experiment simply because the yum face is a subtler thing that's happening inside the animal's mouth, harder to actually measure exactly when it happens. And we're working on that. But more to the point, one of my graduate students now is specifically looking at the output of cortex back down to the area that directly controls the muscles. And it's known that the same set of muscles, or a largely overlapping set of muscles, is involved in the gaping and in the licking that pulls the taste back in. And what we are doing now is gathering data that will hopefully answer the question, what are the exact responses that are getting down to those motor neurons? And if you ask me in like 12 months, we'll know for sure. But our expectation is that it's going to be neurons that are involved in firing high to quinine and neurons that are involved in firing low to quinine, both of which go to that structure. And that means neurons that the neurons that drive the gaping are part and parcel of the neurons that are driving the licking. Thank you.
Thank you for the wonderful talk. It was very interesting to see all the beautiful manipulations you did. One thing you emphasized was the when is important in this representation of information in this population. My question is, how do those neurons know when it is?
So the question is, how do the neurons know when it is? I'm not entirely sure how to answer that simple-seeming question without being challenging and combative, because I think that that it is those neurons that determine the when, and we as investigators are observing it in the activity. But I don't think that those neurons are looking to some source to decide that. And in fact, again, getting back to the question about the variability, I think the system is built to fall into that state, and probably noise plays a part in deciding exactly when that happens. And the neurons are the things that tell us when, and they're not waiting for. They are not looking to someone to tell them. Does that answer your question?
Maybe I can extend my question a little bit. So the early state epoch and the late epoch, the information about if it's sucrose versus quinine didn't get lost in the later palatability period, but there are additional representations of this palatability. Mm-hmm. So somehow these representations are both there later versus earlier. How do we know if there's information or not as a neural system that's looking at this information?
Still not entirely sure that I understand the question, but for sure, when we, for instance, knock out those GC neurons for half a second before the transition gets delayed. So we mess with the processing. And so that means that something is going on that we're not looking at yet. And perhaps this is what you're getting at. We can say, okay, at this time, around this time point, the neuron starts coding quinine. And then at this time point, the neurons say, okay, get it out of the mouth. your question might be, what's happening in between those two that is sort of the drive toward that behavior? How is the system deciding? And if that's the question, my answer is we don't know yet.
That is not entirely my question. Maybe just one last time I'll try.
Yeah, try once more. I'm sorry.
In the late stage of palatability stage, does the brain actually still know what taste it is?
Oh.
And does that representation change?
Yep. So, okay, I'm going to try again. And so, certainly when we get into the palatability state, the information about what taste is what is still there in those responses. And so it does not seem likely that the animal has forgotten. And furthermore, we can do experiments, and I think that there have been experiments done here in which the animal is forced to wait before it does an identification task, where the experimenters actually say to the animal, the task now is, tell me what taste I put on your tongue. Which, for those of you who are not used to work done with non-human animals, isn't trivial, because you can't ask the rat, hey, what did I just put in your mouth? And so the way this task is done is you give the animal a situation in which it gets the taste, and it's told, if it's quinine, lick here for water. And if it's sucrose, lick here for water. And what you can do in these experiments is you can delay that decision. And so experimenters, including experimenters in my lab and experimenters at Stony Brook, have in fact delayed that decision as much as a second or a half or 2 seconds, and the animal is still able to make that decision. And in fact, in that situation, even if the animal doesn't like the taste, it basically waits to make the decision that's required. So clearly, that information is still there. Having said that, we haven't actually done our recordings in that situation. And so I'm not 100% sure that we didn't also change exactly the way the activity works. And so I don't know exactly whether or not you get the transition and then the animal makes that decision, makes that behavior. And so I guess my answer is a little bit hand-wavy in that I can say, we know that you can wait a couple of seconds and the animal still remembers and knows what the taste is. There is still that signal in there. But we haven't done the appropriate experiment yet to demonstrate that you get the transition, and if you then ask the animal to make the decision, it does. We think it will, but we don't know for sure.
Thank you for indulging my questions.
Thank you. Yes? Is there representational drift from? Interesting question. There has recently been a very high-profile paper in general on representational drift, which means, simply speaking, that neurons don't necessarily respond the same to the same things across hours or days. We actually have done several experiments in which we've recorded across a couple of days, and we see something that looks like representational drift, but to be fair, we're actually doing something to the animal in that time period. And so we wouldn't call it drift, we would call it changes with experience. Even if that experience is just having the tastes to try, that does change the situation. It changes the animal's behavior. And it certainly does cause a drift in responding. These neurons no longer respond the same. What doesn't change is the fact of that dynamic. And so a couple of days later, it might be that neurons that were responding strongly to quinine some of those neurons are no longer doing so. In the meantime, some other neurons might be responding more than they did before. And so it might be that the specific set of neurons that are giving the strong responses and have changed, the dynamic stays the same. You still get that sudden transition to palatability coding where the neurons are, in general, reporting what they want to do with the taste. And so this, which we haven't yet published, is itself subversive because it suggests that the signal that's going down to tell the animal gape now isn't one in which it's a particular set of neurons. The signal can itself be contributed, be delivered by whatever neurons are doing it at that moment. And a couple of days later, it might be a non-identical set of neurons. Thank you.
Okay, so I think we are out of time for today's lecture. I would like to thank again Dr. Katz for the wonderful talk, and thank you all for coming and for viewing.
Thank you, everyone. I really appreciate it.